Ever heard of antibiotic resistance? Yeah, that scary things where your medicine just stop working. And the worst part, sometimes we cause it by not finishing our antibiotic just because we feel better. But please remember, bacteria don't rest. They fight back, adapt, survive, and become stronger. So, here is the big question. Where do we find antibiotic? For my final year project, I decided to go on a mini scientific adventure to find out if the local soil in UiTM Jengka hide antibiotic-producing bacteria. Here's what I did. First, I collect soil samples from different locations around UiTM Jengka. To separate the bacteria from the soil particles, I added sterile selang water and mixed the sample vigorously using a vortex mixer. This step helped release the bacteria into suspension. Then, I grew the bacteria on two types of agar, nutrient agar and starch casein agar. For nutrient agar, the soil solution was mixed directly with test bacteria to observe immediate interaction. I get the result for the first screening. But after some repetition, I still failed to get the result in secondary screening. So, I focus on starch casein agar, which is more selective for Streptomyces species. I allow the soil bacteria to grow naturally on starch casein agar without adding any test bacteria. After 48 hours of incubation, the colonies appeared. A total of 45 colonies were successfully grown. I then selected the most promising one and began identification. I observed their shapes, colors, and texture, followed by microscopic observation and simple biochemical tests, including catalyst activity. From this process, I identified 5 bacteria colonies that show characteristics similar to Streptomyces, which are well known for their ability to produce antibiotics. Now, come to the real test. Can these bacteria actually produce antibiotics? I test them against 3 tested bacteria, Staphylococcus aureus, E. coli, and Bachelor subtilis. I use 3 methods. First is spread plate method, where I mix test bacteria and isolated bacteria in SCA. The second one is cross-streak method, where I cross the isolated and test bacteria on the same plate. And the third one is disc diffusion method, where I place a paper disc socket with my isolated on top of the test bacteria. Then I wait 48 hours. If the isolate produce antibiotic, it would create a clear zone where test bacteria could not grow. That's called a zone of inhibition. The bigger the zone, the stronger the antibiotic. And guess what? Some of my isolate did produce strong zone of inhibition, especially against S. aureus and B. subtilis. That means, yes, the local soil bacteria can fight harmful pathogens. I also ran controls to make sure the results were valid. Positive control, Streptomyces, were as expected, and the negative control, sterile water, no effect. The experiment was successful. Based on the result, the zone of inhibition was larger in Staphylococcus aureus and Bacillus subtilis compared to Escherichia coli. This difference can be explained by their cell wall structure. S. aureus and B. subtilis are gram-positive bacteria which have a thick peptidoglycan layer but no outer membrane. This makes them more vulnerable to antibiotic penetration. On the other hand, E. coli is a gram-negative with an additional outer membrane that acts as a barrier, making it more resistant to antibiotic. To confirm the identity of the antibiotic-producing isolate, I conducted further tests. These include gram staining, catalyst test, starch hydrolysis, and indole test. The result consistently matched the characteristic of Streptomyces species. This finding is supported by previous research by Zhu et al. 2016, who reported similar biochemical profile in antibiotic-producing Streptomyces isolated from soil. Therefore, based on morphology, biochemical characteristic, and literature comparison, I concluded that the isolate belong to the Streptomyces genus. From soil to microscopic warrior, my project proved that big solution can come from small things if we know where to look. I am Nazu and this is my final year project, isolating, screening, and identifying antibiotic-producing bacteria from the soil of UiTM Jengka. Because science is not just in lab, sometimes it starts under our feet, soil.